This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 552, recorded on June 14th, 2019. I'm Vincent Racaniello, and you are listening to the podcast all about viruses. Joining me today from Fort Lee, New Jersey, Dickson Despommier.
Good afternoon, Vincent.
How's it in Fort Lee?
It's wonderful over here. It's really nice.
I bet it's the same as here.
Well, I can almost see your house from here. I mean, I'm looking through the arch of the first stanchion of the George Washington Bridge, and I can see the former building that I used to work in.
It's not my house, Dickson.
Now, well, you live more there than you do at home. I know that.
Might be. It's 21 Celsius, partly cloudy. It's a nice day.
Yep. But it's a beautiful day out there. Yeah, it is.
So joining us from Western Massachusetts, Alan Dove.
Good to be here. And it's 71 Fahrenheit, 22 Celsius, and it's mostly cloudy. It's very convective. Lots of cumulus clouds piling up, and I think we might get an afternoon thunderstorm.
A bumpy air ride, Ellen?
Yes, it would be a bumpy day to fly.
Convective.
That's it. Convective, that's right.
Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. We got on my back porch, 88 Fahrenheit, 31 C. Partly,
hot.
Nice.
Partly, yeah, but, it's actually nice. I was sitting out there, partly
It's a dry
sunny.
heat.
I was watching the birds at my squirrel feeder, and
Nice.
It's all good.
Any unusual ones?
Birds? Nah, they're all commonplace, but they're cool. We got goldfinches, house finches, the occasional cardinal. That's about it.
And from Madison, New Jersey, Brianne Barker.
Hi, everybody. It's pretty much the same in Madison as it is in Fort Lee and in the city, although I think we have a few more clouds. Pretty cloudy over here right now.
That's pretty cloudy here, too. Yep.
And we have in studio a guest who's been busy. I don't know. What are you doing? Recording, texting, selfieing? Anyway, she is a former student in my virology course who, I don't know why she knew about TWiV, but she said, can I check it out when you record? So here she is, Kiki Warren. Welcome.
Hello, everyone, and obviously famous professor. Everyone knows about TWiV.
Really?
Everyone does. We
Good
talked
Lord.
about it after class.
We do?
Yeah.
Oh, cool.
And what do you say? This is the important part.
I don't know if she
Fanning
wants,
the egos over
I
here.
don't think she wants to say. But
We're all big fans.
She's sitting there doing selfies, right? It's pretty
I'd
funny.
love to be a phage on the wall.
A phage. I have plenty of phages here. So you graduated this past month, right?
Yeah, from the postbac program at Columbia.
So listen to where Kiki went to college, everybody. Tell us.
So I went to Balliol College, Oxford, where I studied biology and had some microbiology courses as well. None as great as the one I just took.
Oxford, that's pretty cool.
Yeah.
You don't have
Very nice
you don't have
Very cool.
you don't have a British accent.
No, born in New York, raised in Connecticut, and went to England for fun, got more American, but lived in a castle. So
Okay.
Right.
check
Did
that off the
you-
list.
So,
That's cool.
so you said you just finished the post-bac pre-med program at Columbia?
Yeah, it was
Oh, congratulations.
tons of fun.
Yeah. My wife
It's not
did
an
that.
easy one either.
No, but it's got good people in it and some great professors.
So your wife did that, Alan? I didn't know that.
Yeah, Laura was a comparative literature major at Princeton.
Uh-huh.
And then finally admitted that she really wanted to be a doctor. So after that, she went and did the post-bac pre-med program at Columbia and then med school at Columbia.
And now she's a doctor.
She
Yes, she
was
is.
at Columbia?
Huh?
Really?
It's the dream.
Yeah.
She was one of my students.
Yeah, she actually, I met her before she got into Columbia. I met her when she was a lab tech at Mount Sinai.
I'll be darned.
So you want to be a doctor, right?
Surgeon, maybe.
Oh my gosh.
That's not a doctor.
Why?
It feels like the art form. I mean, to be fair, I'm not really an artist per se, but it seems like it's arts and crafts with the body.
A little bit. You know,
The
if you
good
go
part
to
is, yes.
the entrance way to the Columbia's medical school, the old medical school, not the building that's down the block from you now, but it says Columbia College of Physicians and Surgeons.
Hmm.
So in the old days, they used to differentiate between physicians
That's still
and
what
surgeons.
Isn't it still that? Isn't it still PNS?
It is.
It's still PNS.
Still
Yeah,
PNS.
Sure it is. Moshowitz used to call it the College of Fishes and Sturgeons. Did he ever tell that to you?
You mentioned it once or twice.
Yeah, fishes and sturgeons. My dad was a surgeon.
What sort of surgeon?
Chest surgeon.
So he just put his hands elbow deep, I guess.
You know, he
Held the heart.
got sick and couldn't work anymore. He had cancer, and he said, "You know, I'm glad I don't have to be up to my elbows in blood anymore." Although I think that was just him saying because he couldn't work. You know that. But if he had lived,
athletes,
I would
they don't like
suspect
doing it anymore.
if he lived longer, he would have gotten AIDS probably because they didn't know anything about it. Right?
Gloves, yeah.
No,
Oh.
and there was blood everywhere, right?
What
But-
So
Laura
Vincent-
actually
Vincent-
started out as a surgeon too.
Yeah, Vincent, one of your favorite TV shows, and you don't watch that many, is Endeavor.
Hmm.
And Endeavor is filmed in Oxford.
Oh,
Is
Yeah.
it? Do you know what college?
Yeah.
Sorry, we got some rivalries going on over there.
There are a couple of interesting. There was and this is the last diversion. I'm afraid, Kiki, this is a big mistake bringing you here.
No, it's
There's a guy who wrote a series of books about a detective based in Oxford, and they made a TV series out of it, and Oxford Detective series.
Moore? Detective Moore?
Well, there's Morse and
Morse,
there's Lewis
Morse.
and there's Endeavor. There's three of
Oh,
them.
they're all
Right.
related, right? Yeah.
Yeah.
Morse was the first, then Lewis came after, and then Endeavor. Okay. Yeah,
In terms of production, but Endeavor, in fact, is a prequel for Morse.
yeah. Right.
Their Oxford is a lot more exciting than my Oxford was.
Did you
And
like going
The new season starts this Sunday. Are you pumped?
no, I have a grant to get in.
Oh, man.
Like we have two big ones. I'm kind of scared. Did you like Oxford? Was it really interesting to?
Yeah.
It's different, right? Because you spend a lot of time in small groups.
Yeah, you have tiny classes. I mean, my finalist year there, I'd be with 10 or 12 people at most, and then you'd have tutorials with 1 to 3 people. It was awesome. And after, I went to boarding school in Connecticut called Hotchkiss, and after Hotchkiss, it was a fun difference in studying style, which, I mean, it was incredible. It was incredible.
Was that your idea to go to Oxford?
Yeah. My mom very much did not want me to go there. She wanted me to stay in the U.S.
So you did the right thing.
Yeah, disobeyed the parents, like all good teenagers do.
The problem with small groups is that the instructor gets to know every one of you.
Yeah.
And there's no way to hide. There's no way to hide.
Why would you want to hide?
I think that's a great idea. I wish we did that here.
Some people want to hide.
Because I get this to know maybe 10 students in the class who basically who come to office hours, right?
I hear you.
That's the perk of the small liberal arts college.
Yeah. Although I must say, you didn't come very often, but you used to walk with me to the subway after class and continue talking then. And I told you every year there's one student who starts that. They walk from class to the subway. This year it was you. And I remember Kingsley one year who did it. And
So I'm not special.
yeah, I'm sorry, you're not special.
Well, he has to work his way through the
I
cadence.
thought maybe they had an arrangement where somebody always walks the old man to the subway.
That's right. Protect you. That's right.
Exactly.
Okay, before we start talking about science, if you like this show, consider supporting us. Microbe.tv/contribute. A couple of ways you can do that. Give $1 a month, and that helps us. We have expenses. We'd love your support, and it's pretty cheap, $1 a month for not just TWiV, but all the other podcasts. We have a follow-up from Trudy. Dear TWiVers, I'm writing this email in follow-up to your recent discussion about the attachment of polyethylene glycol to a compound, a process that is also known as pegylation. Polyethylene glycol is a very common excipient. Excipients are therapeutically inactive substances that are added to drugs for various purposes, such as for increasing drug absorption, stability, and solubility. Polyethylene glycol in particular increases drug potency by hiding the drug from the host immune system, making hydrophobic drugs more water-soluble, and slowing drug clearance from the blood. But you already know all this. The main
now. I didn't.
That's right.
The main point I want to make is that the pegylation of drugs is so routine that in the patent field we add it as an optional excipient in a boilerplate section to all patent applications for novel pharmaceutical compositions. There are currently dozens of FDA-approved pegylated drugs on the market. But of course, this would never convince an anti-vaxxer of its safety. Best regards, Trudy.
Well, the radiator will never freeze in the wintertime.
You routinely pegylate your patents.
What was this in reference to? Remind me, what was being pegylated?
It was the novel antigen for flu, where they were covering up the immunodominant epitopes.
So we were worried that maybe it would be difficult to get to use it, but obviously not. So many things are pegylated. No. All right. So today we have a snippet which is very interesting, and maybe Kiki will remember some of this because it has to do this every year in my virology class. I always, when I teach about DNA synthesis, I always say all DNA polymerases require a primer. Do you remember that?
Remember it being mentioned, yeah.
You remember it being mentioned. And so, DNA polymerases are enzymes that copy DNA. We have them in our cells. Many viruses encode them in their genomes, or they take advantage of the ones encoded in the cellular genome. And so they're really important. They have to copy the DNA, and there's an enzyme called DNA polymerase that does that, and then there are many other accessory proteins that are needed as well. And we have always thought that they require a primer being a small nucleic acid that hybridizes to the template DNA, and the DNA polymerase can latch onto it and begin copying. And of course, the most famous primers are the small RNA primers that initiate DNA replication in many organisms like us. And so we have two papers today that show not all polymerases require a primer. So two papers today get rid of this dogma. There are two polymerases where we don't need a primer. Now, this whole idea that you need two enzymes is really interesting. Actually, the primers are made by enzymes called primerases. And they sit down on the template and they make a little RNA, typically an RNA primer, by copying the template. So they're initiating de novo, which is what we say when we don't need a primer. But then they leave, and the main DNA polymerase comes in and takes over. And one of these papers says, well, why do you need two enzymes? And that's something that we'll get into, but I want you to think about it in these terms. It's not easy to initiate and to elongate, right? Because elongation, you're like a race car. You're going down the track, you're zipping down the template, you're churning out a copy, you're going really fast, right? But think initiation, you got to get to the template, you got to put a base down. It's a little wobbly because it's just one base, then you have to put a second and a third. It's a totally different thing. And so probably that's why, and at least in my view, that's why you need two different enzymes.
I think also when you're polymerizing, if you're going to do it right, you can't let go. Okay,
right.
You got to be accurate, and you can't let go. You have to synthesize really long stuff. You can't make any mistakes, and you got to keep going for thousands and thousands of nucleotides.
That's right. Now, you may be wondering, what about other enzymes like, let's say Pol2, polymerase 2, which copies DNA, makes mRNA. That does not need a primer, but it's really hard because it often fails. Because when it makes the primer, when it starts initiating, it goes pretty slowly and it often falls off. And once it's made, 20 or 30 bases, then there's a major conformational change so that it can now become a speedster and go zipping down the template. But
it shifts gears.
Yeah. Mm-hmm. But they fail a lot because that initial transition from the slow business to the fast is hard. And that may be why DNA polymerases, have their two enzymes to do this. But
yeah. I think that when I was reading this, I was like, oh, I know of a polymerase that doesn't need a primer. And I hadn't really thought about that difference, that the polymerase I was thinking of usually adds a couple base pairs, and that difference in terms of having to polymerize a really long stretch.
Right.
Yeah.
So important.
Yeah, actually, these papers got me to think about it in those terms because I never really made that connection. And in RNA polymerases that copy RNA genomes, some of them need primers, some of them don't. If you don't use a primer, you need to have something for the enzyme to latch onto. And so that many of these polymerases, they put a little piece of protein in the active site so that the first bases can set up against it, and then it goes away once polymerization begins. So there's some unifying things here.
Vincent, did you say you can't get to first base unless you have a primer?
No.
That sounds like a baseball team.
Not a primer. Yeah, Yogi Berra would say that, right?
Right. Exactly.
Nobody primes there anymore.
I guess not.
It's too popular. Nobody primes there anymore.
That's right. If you see a primer, take it.
The active site, at least for some of the RNA-dependent RNA polymerases that don't need a primer, there's a little piece of the protein that sits there as kind of a backing for that first base that comes in. Otherwise, it would slip out. And then after maybe 20 bases are put in, that piece of the protein moves out of the way, and then the regular polymerization can begin. So it's kind of like the conformational change that happens in Pol II when you're going from initiation to elongation.
Right. So none of these though are DNA-directed DNA polymerases.
No.
Right.
So what we have here are two papers. One we'll focus on is a phage enzyme, and the other's in mobile genetic elements. So the phage paper is called Deep Sea Vent Phage DNA Polymerase, specifically initiates DNA synthesis in the absence of primers. So this is published in PNAS. This is actually a couple of years ago, 2017, but I just discovered it last year, and I wanted to bring it up. And it comes, the first author is Bin Zhu, and this is from Charles Richardson's laboratory. He's the senior author here. And there are a variety of places. So Richardson's at Harvard Medical School. There is a Huazhong University of Science and Technology in Wuhan, the Japan Agency for Marine Earth Science and Technology.
Charles Richardson is now 83 years old.
Wow.
He's been at Harvard Medical School forever. And I'm familiar with his work because he spent most of his career figuring out DNA replication in bacteriophage T7. Okay? So he's made a whole career on bacteriophage, which is a good trick, but he does just amazing biochemistry.
Yeah, it's quite clear.
The penultimate author on this is Stanley Tabor, who has an interesting history because he was a graduate student in Richardson's lab in the 70s and hung around and has been working with Richardson ever since and shows up on a very large percentage of his papers. He's independent in his own way, but has grown up with Richardson doing this stuff.
So they write right away, a long-held dogma was that DNA polymerases are unable to synthesize DNA de novo. Primer is needed. And then they say, why do we need two polymerases? And it goes into this argument about how elongation is different from priming that we just talked about. So the subject here is a virus that infects a bacterium that lives down by the deep sea vents.
Which is
in the
cool
deep sea or near it?
Deep sea vent. No, by the deep sea vent. It's called deep sea vent bacteria, epsilon proteobacteria.
But did you realize that a lot of those bacteria are actually coming out of the vent?
Right. I don't know exactly how they sampled this.
And that's very, very hot.
Yes, I think the epsilon proteobacteria are in the vents.
They're in the vents?
Mm-hmm.
Yeah, that's where their polymerase comes from that's used in the PCR reactions now, right? Because it's got such a high temperature stability that you can use it very fast for cooling and heating and cooling and heating. That's why it works so well.
So this is the first phage that infects these deep sea vent bacteria.
And that's the food for the rest of the food chain in deep sea vent.
It's called NRS1.
Wow.
All right. And they had the genome sequence, and they looked for genes involved in DNA replication. And they see a helicase, they see a single-stranded DNA binding protein. These are proteins that you need that are involved in DNA replication, but they didn't see a polymerase. They didn't see what they call a replicative polymerase. That is the polymerase that does the long copying of DNAs. But what they did see is some protein with a little bit of homology to a primase, which is the enzyme that makes primers, right? So they said, maybe this is a weird polymerase. So they took the gene, they made the protein, and they did some biochemistry. They purified the protein, and they have a nice little synthetic template, and they have a primer, and this protein makes DNA, so it can do polymerase. But if they leave out the primer, it still copied the template. And their control is T7 DNA polymerase, which is dependent on a primer. Right? So it will make what they say robust DNA synthesis in the absence of a primer. And it's quite clear. And they try different templates, and they see there's variation in ability. And it turns out that this polymerase, they call it now a DNA polymerase, recognizes a specific sequence in the template to initiate synthesis. And I don't think that's a feature of most polymerases, right, Rich?
No, not DNA polymerases, certainly the ones that require a primer. And I don't think it's a feature of primases
either.
No, I don't think so either.
This is unusual in this regard.
So they're brought there by, they can be brought there by DNA binding proteins, right? Mm-hmm. Like I know for viruses anyway, like T antigen helps bring the polymerase to the DNA. Yep. Mm-hmm. So this polymerase seems to initiate at this. It's an 8 nucleotide sequence, and it initiates there and doesn't need a primer, and it does pretty well. Makes pretty long products. And it actually, they do some mutagenesis of the gene, which is quite nice. And they can see 2 domains. There's an N-terminal site that seems to have the active site of the enzyme. And then the C-terminal site seems to increase what we call processivity, the ability to make long DNA. And the synthesis begins with a C. They do a series of experiments where they leave out each one of the bases and seems to initiate with a C, a single C residue. So this DNA polymerase can just sit down on the template and start polymerizing when the first base is a C. And it associates with, they show that the helicase and the single-stranded DNA binding protein encoded in the genome associate with this enzyme and help to make the DNA. So the helicase is unwinding the duplex, right, as the polymerase is going down there. So that's cool. That's the result. This is a phage polymerase that doesn't need a primer. And so they say that they originally picked it out by homology to the primase, and they said, but it's not a primase. It has different features from a primase. Mm-hmm. And one of the features of a primase, there's this zinc binding motif that helps it recognize the template. There's no zinc binding motif in this polymerase, so it's different.
It also preferentially incorporates deoxyribonucleotides, if I've got this correctly.
That's right. Yeah.
Whereas a classic primase does ribonucleotides. Yeah.
I think this one will take NTPs, but it prefers DNTPs, right? Yep. So this is a cool part of their discussion. The polymerase has features of 3 polymerase families. DNA polymerases, right, because it can copy DNA. Primases and RNA polymerases, where you don't need a primer, right? And I think I think they showed that the enzyme has a little bit fits and starts when it first begins, right? It falls off and makes abortive product, just like RNA polymerases.
Right.
Yeah. And
so they say, wow, since we found this at the bottom of the ocean where there are old bacteria, maybe this is like an ancestor of all current polymerases, right? Mm-hmm.
Maybe this dates from the transition from RNA world to DNA world.
Yeah, that's right.
They're not archaeobacteria?
No, they're extremophiles, but not archaeobacteria.
Right. Not so the other question I have is, at what temperature did they carry out these reactions?
The temperature optimum for the polymerase is 50 degrees.
Look at that. Look at that.
Yeah, good question. Not 37.
No, not 37. No, that's right.
And if they feed it, they also clone a couple of other accessory proteins, a single-stranded DNA binding protein and a helicase, to throw in the reaction to see if they could add processivity to it, which they can. And they give it a small mini circle so it can do what's called rolling circle replication, where it goes around and around and around and peels off a big, long strand of DNA. And it
also
looks like they
toilet
get
paper
stuff.
replication.
Yeah, looks like they can get stuff that's up to 8 kilobases. Is that right? No, that's nucleotides. It's in excess of 1,000 nucleotides. Okay, so pretty big.
Yeah, that was the impressive part to me. But 50 seems a little low compared to the temperatures we use in PCR.
I was going to say, that's right.
Yeah, I think it's hotter down there in the vents.
Well, but it's important to remember that. Okay, but at the deep sea vents, there is a really extreme temperature gradient.
Yeah, that's right.
Because the water is near freezing,
right,
and the vent is boiling, and then over a very, very short distance, you have this transition of ecosystems, really, where you've got the extremophiles that have to be at in the 90s Celsius and then down to stuff that can survive at much lower temperatures. So this may be from somewhere in that gradient.
Also, it's nice if you don't have to go above 50 degrees, right, in your water baths.
Oh, yeah.
Because then you have to use polyethylene glycol.
There you go.
Is that
or
the
you have to
antifreeze?
replace the water a lot because it evaporates.
No, the antifreeze is ethylene glycol, right?
Ethylene glycol, I think.
Right.
Yeah.
And
we used to use glycerol. We had a bath that was 50% glycerol that we could get up to like 110 degrees.
Glycerol.
That was for doing denaturations.
Yeah. Yeah.
But PEG is used to preserve things also. There's a ship that sank during its maiden voyage in Sweden called the Vasa.
Yeah.
Heard of it? And they when they recovered it, it was intact. Nothing was damaged at all because it sank in fresh water, so nothing attacked the wood. But when they got it above the atmosphere, it started to oxidize the wood, so they covered the whole thing in polyethylene glycol.
Well, this is why sea captains in the old days often had pegylated legs.
That's right. Exactly right. Our mateys.
So in anticipation of the other paper that I assume we're going to mention on this, I want to make a comment about this enzyme. Most DNA polymerases, or at least the classic ones that were taught about in the dogma, have a proofreading function.
Right.
So it's an exonuclease. So effectively, if the polymerase puts in an incorrect base, it can effectively back up and clip out that base and then try again to put the right one in. And of course, for DNA synthesis, proofreading is really important because getting the code right for subsequent generations is really important. This isn't as important for like messenger RNA synthesis. You're just going to throw it away anyway. A couple of goofs, it doesn't matter. But we're talking about heritable sequence here, so you want to make sure it's right. That domain that does the proofreading, or at least the exonuclease domain that clips out the wrong base, is usually a separate domain on the polymerase. Actually so separate that in coli polymerase you can actually cleave it off with an appropriate protease. This polymerase does not do that. It does not have an exonuclease domain. Okay, so they don't make a big deal about this. They test it for exonuclease activity. They don't comment a lot on its proofreading capability, but what I take away from this is that this enzyme can't proofread and doesn't have the exonuclease domain. And I think that's relevant for the next paper.
It's probably also, this is a phage enzyme, right?
Yeah.
So a little more error tolerant, or a little more permissive to errors. You screw up your phage genome, you've got a billion other copies of it floating around, and this is how you evolve.
Maybe.
Right.
Or there could be another protein that's involved, right?
It could be an error, a proofreading program.
Because if this is an ancient polymerase, maybe we haven't got around to combining or having it. The one of the interesting things, I think it's in the second. what they don't show here is that this enzyme actually replicates the viral genome, right?
That's right.
Right.
That's important. It's all biochem. It's all in a test tube.
So,
and actually, a couple of asides on this phage, because I looked it up because, I just get into this. It's a temperate phage, so it can integrate into the bacterial chromosome and then pop out and replicate. And the genome is about 37 kilobases, 37,000 base pairs long, and that's enough to encode about 35 proteins. So it's a modest size phage. And just for kicks, it's worth mentioning that the genome is, I don't know if we've talked about this before, terminally redundant and circularly permuted. Okay. What that means is that, and the terminal redundancy is there's sequence on one end is repeated on the other end of the linear chromosome. And it's a direct repeat. So if it's ABC, and then you go through a bunch of other sequences and you get down to the end in the same orientation, it's ABC again. So it's a direct repeat. Circularly permuted means that if the sequence is A, B, C, D, E, F, G, some genomes have A, B on either end. Some might have D, E, F on either end. Okay. And the way you get that is to replicate DNA by this the toilet paper model that we talked about before, the rolling circle, and you make big concatenamers. So you have one linear genome after another. And then the hypothesis is that the way you package it is to stuff it into a head of a phage and then clip off the end and then stuff in another.
Are you stuffing toilet paper in a head?
Yeah,
is that what you're doing?
Okay.
So.
And if the head is a little too big, you can stuff in a little more than one genome's worth and cut it off. So you get this direct repeat, but you're starting now in the next head in a different place. That's where you get the circular permutation. I've just thought that was just so cool my whole life.
Yes.
So, sorry, I had to rant.
No, I figured you'd like this, man. You're a T7 guy, right?
Yeah.
All right. So there's one other thing, but I think it's mentioned in the second paper, so I'll wait. The second paper came out around the same time. 2017,
just a few months later.
Primer independent DNA synthesis, and this is actually the first one that I noticed. Primer independent DNA synthesis by a family B DNA polymerase from self-replicating mobile genetic elements, and this comes from. It's very interesting. One of the authors, Margarita Salas. So there are a number of authors here who I know. She is from. Universidad Autónoma in Madrid. She was actually a reviewer of the first paper. And then we have some people from the Institut Pasteur. This is great. The Unité Biologie Moléculaire du Gène chez les Extremophiles. It's the name of the department. Pasteur, Max Planck Institute, Delft University. First authors, Modesto Redrejo Rodriguez. And then we have Patrick Forterre, who has published a lot of papers on the origin of viruses, say, Margarita Salas, and Mart Krupovich from the Pasteur, who, when I was president of ASV in 2015, I had him on the morning program where he talked about other things. So here they did a blast search against the entire bacterial genome database, and they looked for polymerases. They seeded the search with a polymerase B from a bacteriophage, and they ended up finding hits which were on mobile genetic elements, and they're in many bacteria, and they're also in mitochondria. And these mobile genetic elements encode polymerases, which are called pipolins, Pipolin. Pipolin.
Where does that come from?
Oh, I looked it up when I first read it. Pipolin. Kiki's looking it up right now.
I feel like it's primer independent and then Paul.
Okay, yeah.
But I think they didn't know that when they named pipolins, right? Because pipolin is not new to this paper. Primer independent is, yeah, I guess it is. We refer to these elements as primer independent polymerase B encoding mobile genetic elements, pipelins.
Okay, so they named
maybe like integrated.
Maybe because some of them are integrated. So the pipelin is actually the element. It's not actually the enzyme. So I got that wrong. Pipelin is the mobile genetic element. Anyway, they found these polymerases on a variety of mobile genetic elements, some of them integrated into bacterial genomes of many different phyla. Some of them are on plasmids. And also they found them on mitochondrial plasmids. I don't know if everyone knew this, that mitochondria have plasmids. Yep. They have circular DNAs in them, and they encode.
There's DNA everywhere.
There's DNA
really
everywhere. It's a real problem.
Yeah.
It's exactly a problem. And so they find these polymerases on these mobile genetic elements. And so they say, let's make some of these proteins and see what they do. So they produce the protein, they purify it. They took one of them from E. coli, one of the pipelins. That's just the pip, isn't it? You guys don't know that phrase.
Yes, I do know that.
You must know that from the UK,
just letting
right?
it sit there.
No, you don't know it? Okay. So they purified it, and they did polymerase assays, and it can copy DNA. It's a polymerase. And guess what? It doesn't need a primer. Same idea. And they compare it with primer-dependent enzymes. Clearly, it doesn't need a primer. So here we have another example, and this is not a phage enzyme. Of course, this is in bacterial and mitochondrial genomes.
So if any of the authors are listening, were they already working on this project before Margarita Salas reviewed the previous paper, or was it inspired by that?
Good question.
It's got to be before, because they're published only a few months apart. And there's a lot of work in these things.
Yeah, there is a lot of work. The first one was published in March, and this one was published in November.
Right. So they have a couple of interesting things here. First, they say that probably these are really old. The pipelin, the genetic mobile elements in the bacteria are probably as old as the bacteria, like 2 billion years old. And they say that probably these elements went into eukaryotes along with the mitochondria, and the mitochondrial polymerases are descended from them, which is cool. And that's sort of the same thing we talked about when we talked about these mitochondrial viruses. They probably came from the bacteria that were before mitochondria. And somewhere in here, I think they talked about this. So these enzymes, so all these nucleic acid enzymes have a palm. They look like a right hand, palm, fingers, and thumb domain. But these don't. These just have a palm. They don't have fingers and thumb, which is interesting. The fingers and thumb often make channels in some enzymes and others not. But these don't. There's just a palm, which is the active site, which I thought was interesting.
And an opposable thumb is a characteristic of all primases, right?
That's right.
Arg.
And I rewrote the RNA synthesis chapter for the textbook. And there I learned now we have so many RNA polymerase structures. The picornaviral polymerases are the smallest. They have a palm, a thumb, and a fingers domain. And they're thought to represent the core polymerase. And all the larger RNA viruses, they decorate that with additional domains that have different functions. But it's clear that there's even a more basic polymerase, which consists of just the palm itself.
Right. No
fingers and no thumb. So I thought in terms of evolution, that was pretty interesting. And then the last thing, they say these enzymes, PyPol-B, has great promise for developing novel biotechnological applications. And they say it's really processive, and you can get whole genome amplification. And there's no sequence requirement for initiation. So this could be really good for amplifying entire genomes relatively quickly. So that would be interesting to see.
That was something that occurred to me with both of these papers is, primer independent enzymes. Hey. So you don't have to seed things with random primers and hope that you get amplification.
Mm-hmm.
Maybe you can turn these into useful lab tools.
So for now, if you want to sequence genomes, you often amplify them with random hexamers that prime everywhere, but you don't have to do that anymore. Maybe if you can get enough product from this, that could be really interesting.
or if you can take pieces of this and attach it to your good processive PCR enzymes. I mean, you can do all sorts of things here.
Yeah. It's a good time to be a molecular biologist.
Sure is.
It is.
So I want to contrast.
Not a doctor, man.
I want to point out a couple of differences between these two enzymes. First, as we already mentioned, I don't know quite what to make of this, but this one does not require a sequence.
Right.
It's sequence nonspecific, whereas the phage enzyme recognizes a specific sequence, which actually occurs twice in the phage genome, right next to the gene for the protein itself, which may be of significance in terms of the evolution of that phage. And importantly, this bacterial enzyme, this mobile genetic element enzyme has a proofreading function and has a proofreading domain, whereas the other one does not. And then they have this paragraph in their discussion that really caught my attention, where they say, although in certain virus or plasmid encoded proteins, the prim-pol domain is fused to various helicases that can synthesize large DNA products, and they reference the phage paper that we just did. These enzymes lack the exonuclease domain, and their DNA polymerization on longer templates appears to be mainly distributive. That means the enzyme will come off and come back on, okay, rather than one big long chain. They say, thus it is generally considered that the role of prim-pol proteins in vivo is largely restricted to the synthesis of short primers, which are extended by the cellular replicative DNA polymerases. And they reference a couple of other papers, not the one we just did. So, and that's their only reference of the phage paper. So it's the implication, it seems to me, that they are implying that they don't think that the phage enzyme is actually the DNA polymerase that does the phage genome, which goes back to our discussion that the one thing missing from that paper is showing that enzyme actually replicates the phage genome in the cell. Their implication is that, it's got these properties and that's cool, but it's probably mostly there to make primers, and that maybe a cellular DNA polymerase finishes the job. I'm not saying that is the case, but yeah.
But
right.
these
because if you can initiate without a primer and extend a little bit, you've installed a primer, and there are plenty of other enzymes around that can finish the job from there.
Right.
But their enzyme, the PIPPOL-B, that's probably just replicating the mobile element.
Mobile element,
right. So, does this really then violate the dogma or not? Or is it still true that replicative polymerases are primer dependent?
Maybe.
Doing entire genomes, phage genomes, or eukaryotic genomes.
I would say that we don't know yet.
Yeah.
Okay. They got
We have not yet found an enzyme that we know is a replicative polymerase that does a whole genome that starts without a primer. But we've got a couple of leads.
Yeah.
As they say, there's a lot of new sequence out there of viruses and hosts, so it just has to be looked in. But it may be that to do an entire genome with one enzyme doing priming and extension, it just doesn't work because it's too big. And, doing processive replication for that many thousands of bases may not be compatible with also initiating.
But even if it turns out that this phage enzyme is not actually the replicative enzyme, it has some really unique properties. The sequence specificity on the one hand, and the fact that it lays down if it's just a primase, it lays down a DNA primer rather than an RNA primer. So it's different.
And what someone can do now is you've got these two additional polymerases that certainly the deep sea vent one didn't look a lot like a polymerase before, but now we know it is. And so somebody can do a bioinformatic data mining as more and more sequence comes in and kind of widen the space of what we consider to be a polymerase sequence.
Yeah, sure.
Yeah. I would be interested to see what they find in microbes that have a much slower replication rate. I would assume that the problem with processivity would be a real problem if you're trying to replicate very quickly. And so maybe if you replicate really slowly, it doesn't matter as much.
Yeah.
My guess is that this is true, that it doesn't need a polymerase to start the initiation process because dogmas arose much later in evolutionary history.
Yes. Well, what I was thinking is that most of what we understand about DNA replication was done using a few model organisms, right?
Mm-hmm.
Yeah. And there's way more out there than a few model organisms. So who knows how many things are ready to be disproven, right?
I was just thinking there's more than one way to skin this cat.
Yeah.
Okay.
Yes. Anyway, I like this a lot because, in virology, not many people study DNA synthesis anymore. It's really there was a time when this was what a lot of people did.
Oh, yeah.
But it's kind of gone away. But I think it could be revitalized by basically this is all based on sequencing. We can do so much now. We can get interesting genomes and explore the proteins encoded in them.
So you think there might be more than 7 ways of making DNA?
I love seeing all the biochemistry.
There's 7 ways.
The biochemistry is just beautiful.
7 ways to get to mRNA. That's the thing, Dickson.
Oh, okay.
Not to make DNA.
All right.
Right. You remember that, Kiki?
Yeah, it was the whole graph.
With the Baltimore scheme.
That's it.
We had all 7. We had to do that for every exam.
That's right. What do you think about this? Is this interesting?
Yeah, it's fascinating. I'm not sure why we're only looking at extremophiles or whether
I think
that's the secret to our own.
That's where these enzymes are.
That's a good question. There's certainly phages in other environments. And this group happened to look at that. But they're also old, so maybe that's one of the things that if you want to look at the evolution of these polymerases, you look at very ancient organisms, right?
Because the proofreading was not accurate enough? Well, there was no proofreading, so you'd want to get rid of that?
Well, it didn't start out with proofreading probably, and then it evolved most likely. That's a possibility. But I'm sure you could find similar things in other environments. I just think they happen to be looking at extremophiles, right?
Yeah, well, I was wondering about that a little bit myself. And in the second paper, they do say that when they use the pi-pol sequences, they found sequences from quite a few different groups of bacteria.
Yes, right.
And those are not all types of extremophiles.
Yeah, that's right. And these mobile elements, so they're quite widely dispersed, But you would probably like to go down to a vent and sample some bacteria, right?
Yeah, it'd be awesome.
I think you're
on the Nautilus.
Right.
Well, there are some in the back of people's minds, I'm sure. There are payoffs that result from discovering unique features of proteins and enzymes, or other enzymes too, that can survive at that temperature and pressure.
Yeah.
I mean, New England Biome, right? New England?
Yeah. Biolabs.
That's right. They made a fortune on that.
So you're talking about money, Dickson, right?
Well, if the heck with grants, I'll just fund it from the money that I make selling this.
Yeah, but I think it's really just interesting and to understand how evolution went and how polymerases evolved. I think that's just great. And next year in the class, now I'm going to tell everyone, now there's some primer independent polymerases.
Yeah.
Although that might not happen in situ, you're saying?
Yeah, it may not happen for whole genomes. We'll see. Maybe by then it'll be sorted out, but I think it's worth mentioning because I do make a point of DNA polymerases need a primer. By the way, I don't know how many students will care, though. We'll see. You should just take the course again and see. Actually, you don't have to actually go. You can just take it online.
It's true.
The other paper is also really cool. It was published in Nature Communications. This one.
So those two were a snippet that took up almost an hour. But let's now go on to another paper.
Well, we could just reverse them and make this a snippet and do it quicker. This one was published recently, Nature Communications. Envelope viruses distinct from hepatitis B virus induce dissemination of hepatitis D virus in vivo. So this continues an arc of hepatitis delta virus, which I don't believe I taught this year. Because we had a snow day or something, and that lecture had to go.
We stopped at C.
That's right. We stopped at C. So a couple of weeks ago, hepatitis delta virus It's a really unusual human virus, 1.7 kilobase RNA genome that encodes only a single protein, delta antigen, and it needs to be in a cell with hepatitis B virus to be packaged as a virus particle. And so this virus has been found in people with hepatitis B largely, exclusively, as far as we know. So it requires hepatitis B virus as a helper to provide a capsid, essentially.
At least that was the dogma.
That was the dogma. Now, a couple of weeks ago, we talked about related viruses for the first time in snakes and in birds.
And we questioned what their helper virus was.
Because there was no hepatitis B virus in those identifications. So one of the things we said is it possible that a different virus might be able to serve as a helper for hepatitis delta virus? And
the
lo
answer?
And behold, the answer is yes.
Yes.
And these people clearly heard us and did the experiments in just a week.
Real quick.
That's right. But they got the grant and we didn't.
Oh, boy. Yes, you're right.
Especially, this was published at the beginning of May, so they actually anticipated the question.
Before we did it on TWiV.
Yeah.
So these authors are from a university in Lyon, Université Claude Bernard, Lyon.
Beautiful city.
Never been. And the CNRS in Paris. And the first author is Jimena Perez Vargas. And the last author is Francois Loic Cosset. And I happen to know Camille Sureau. He's been working on hepatitis viruses for many years. So the question here, there's
also Bertrand Boson, whose brother Higgs is very famous in physics.
Yes.
For his particle?
Do you know him personally?
No.
No.
He's hard to find.
So the question is, can you package hepatitis delta in a particle from another virus other than hepatitis B virus? I don't know why no one has done this before.
No, they also note a really interesting observation in the introduction.
Yeah.
That I have lots of questions about, where hepatitis D antigen and RNA was found in some patients with Sjogren's syndrome, which doesn't have anything to do with the liver. It's a salivary gland and eye-related disease. And so why you would see this virus outside the liver and away from hepatitis B was sort of an interesting question.
Very, very important. These patients do not have hep B.
Because of the dogma, probably everybody just blew it off, right?
Right, exactly.
Probably. Well,
but actually, before we get into this for the Calebs out there, I want to say that hepatitis D is important to us because it exacerbates clinically the hepatitis B virus infection.
Right.
In particular, hepatitis B, the acute infection is nasty enough, but it can cause a chronic infection where you have hepatitis B for a long time, which over time can cause liver damage and ultimately liver cancer. And if you have a co-infection with hepatitis D, which because it needs a helper can't replicate by itself, it exacerbates that whole process. In particular, there's the degradation of the liver, and the chances of getting liver cancer are greater. So it's a clinically important pathogen.
And there's a hepatitis B vaccine now, which is getting rid of that virus if people get it, which has been thought, well, okay, we certainly won't have to worry about hepatitis D once hepatitis B is gone, but now we've got to revisit that apparently.
Right. Good point.
Yeah. It may be in way more people than we think.
Yeah.
So very simple experiment. You take two plasmids. One encodes a glycoprotein, and you can do hepatitis B, but they also try vesicular stomatitis virus and hepatitis C virus glycoproteins. And the other plasmid encodes the delta genome. So if you put just the delta plasmid in cells, it will replicate because it's copied by Pol2. And then if you put the hep B glycoprotein plasmid in, you make particles. And you can show they're released into the supernatant, but they also find particles if you put in the glycoprotein of VSV and hep C.
Yeah.
So these glycoproteins are, for these various viruses, the proteins that show up on the surface of the particle embedded in a membrane. So they're important for enveloping the genome and getting out of the cells and into the next cell. They're basically the coat and the
right. So all your envelope viruses have glycoproteins on the outside.
And they do a bunch of experiments to show that, in fact, there are particles being produced and released from the cells. They can be immunoprecipitated with antibodies against the various glycoproteins. There's viral RNA inside of it.
Beautiful bunch of experiments too.
Classic virology.
Classic virology to make it absolutely convincing that these glycoproteins are making virus out of this delta.
They're purifying them in gradients, measuring the density.
Doing
now, another thing to remember about glycoproteins is that these are the antigens that the immune system is going to recognize.
Right.
That's right.
So if you test somebody for hepatitis B, test for the surface antigen and the core to distinguish between whether they've been vaccinated or infected. But hepatitis delta is going to show up as having hepatitis B virus coat. Although, if it picked up a coat from something else, you would assume that it would show up. I would expect that those patients would, if they exist, would not have antibodies against Hep B necessarily, but they might have antibodies against these other viruses.
Yeah.
Mm-hmm.
How many hepatitis letters are there in the?
We got
virus?
A, we got B, we got C, we got D, we got E, and we got G. And I don't know why they skipped that. You could probably come up with a reason.
No, there was briefly, I think the story was that somebody found one, called it hepatitis F, and then it turned out it was a spurious finding. But by then, somebody had already found hepatitis G.
Okay.
And so F just got permanently retired.
I have a more imaginative and colorful reason.
Yes.
And by the way, these are all quite different agents. They're quite different agents. They have in common that they cause hepatitis, and that's about it.
But does hepatitis D serve as a virulence factor for the other diseases that they do cause?
Well, it does.
So you might have an unidentified disease. We don't know what's causing this, but a lot of people are getting sick. They had the other entity, which doesn't make you sick, and then they caught hepatitis D, which then makes you sick because the other ones now can do that.
Don't know.
That becomes a question.
That's a question, yeah.
Right.
That's the question.
Because there are a lot of unexplained outbreaks out there, right?
Oh, yeah.
Yeah, but nowadays you do, in many cases, you deep sequence, right? Mm-hmm. And so far we haven't picked up Delta, but I think after this paper, people are going to be looking harder.
I'll say.
Yes.
For sure. Now, these particles are infectious. They're made with VSV or Hep C glycoproteins. They're infectious. They can bind to cells that only have the right receptors for those viruses. They actually use antibodies against the viral glycoproteins to block infection. So anti-VSV will block the VSV delta, and same for Hep C. They have an inhibitor of the hepatitis B virus receptor, taurocholic acid, which only blocks the B, which is kind of cool. Now then they go,
made in the liver.
What about other glycoproteins? They try a whole bunch of virus, a bunch of retroviruses, including HIV, influenza virus, lymphocytic chorial meningitis virus, paramyxovirus, dengue virus, West Nile virus. So what they find is that a human metapneumovirus, which is a paramyxovirus, dengue virus, and West Nile glycoproteins will rescue delta particles. It worked with LCMV, but not as well. But not with HIV, which is probably good.
Oh, yeah.
Yeah.
It didn't do that.
Can you block replication of hepatitis D with antisense?
Yeah, I'm sure you
therapies?
could. Yeah.
I mean, would that be a potential intervention strategy for people suffering from this?
You can deliver antisense, sure.
Yeah, but they're probably. So right now, a drug against Hep C has been developed, which is an RNA, which actually blocks a small RNA that's needed for Hep C replication. So that can be delivered to patients, but it's an extensively modified oligonucleotide, so it's stable, right? It lasts a long time. Hey, maybe it's even pegylated.
I'm going to ask.
Who knows?
Exactly what I was going to say.
But you could. So one of the cool experiments is that they show this all works in non-liver cells.
Mm-hmm.
Yeah.
So that means this could potentially infect many other cell types, right, besides liver. So
even mosquito cells.
Yeah,
cells.
I really liked that part.
Which they look at because of dengue virus, which I think is just amazing.
Or West Nile.
Yeah, that too.
Yeah, so hepatitis delta seems to be able to just take on the coat of some other virus and sort of assume its identity in terms of going into other cells and spreading around.
There's got to be a title in there, right?
Maybe
taking on the code of many other viruses. They also show that so far we've done this with plasmids, but you can actually take virus, Hep C or dengue virus, infect cells, put in Delta virus, and get out particles, which is what would happen in people or other animals, right? No, it wouldn't be plasmids doing these these infections in people. So it also works for infections.
Yeah.
So dengue and Delta. Can you imagine dengue, which is so prevalent?
Yeah.
Yeah.
That could be a really interesting thing. And finally, so what would you like to see next? How about a mouse experiment?
That'd be great.
So they have humanized mice. So they have human livers. So they can infect them with Hep B.
How do you make those? You know how you make those mice?
Human. The way I make them is I get out my checkbook.
Right.
And then the check bounces.
Call Jackson Labs.
Get 'em.
And
I'm not sure about the liver humanized mice. Would you like to look it up?
Nah.
Oh, here we go. Let's see. So you take human hepatocytes, which apparently you can buy from Corning, right upstate. They intrasplenically inject them into a triple mutant mouse knocked out for fumarate acetoacetate hydrolase, RAG, and IL-2 receptor gamma chain. They're kind of like skids almost, right?
Yeah.
Those are
severely immune deficient.
Yeah.
That triple knockout mouse is the mouse that you use to do a lot of humanized mice.
Okay. So it's not NOD skids. It's different from NOD skid, but it's basically similar. Yeah. So these.
Do they also have a mouse liver?
Yeah, it's a certain fraction of the liver is human.
Yeah, it's not the whole thing. Yeah.
So these cells home to the liver and
they grow.
Set up.
Yeah. And because they're immunocompromised, they don't get rid of them. Yeah.
Right. Okay.
And then you can infect them, and they show that you can infect them with these hepatitis delta viruses made with what did they use here? They used Hep C. So these mice will support Hep B and Hep C infection, and the hepatitis delta made with either Hep B or Hep C will infect these mice, and you get a viremia.
Yeah.
So it makes viruses in animals as well. Quite interesting. Lots of interesting questions. Like how do these deltas get packaged? If you remember, in order for Hep delta to get packaged in the Hep B envelope, here's where we ran into trouble before. The delta protein has to be farnesylated. Mm-hmm. Remember that? I know Johnny had a little trouble with that. So we put a farnesyl group, which is a lipid, onto the delta antigen, the delta protein. That allows it to get incorporated into Hep B. But these other glycoproteins apparently don't work that way. So there has to be some other mechanism for getting, which is really interesting that it would work in more than one way. Other determinants of envelopment, they call it.
Yeah, they have a long discussion here contrasting the viruses that don't envelop delta with those that do, trying to figure out whether it has something to do with the mechanism of envelopment. Mm-hmm. Okay?
What
they can't make a totally clear story out of it, but clearly that's where the thinking is going.
Yeah, so hepatitis D then seems like almost a meta-parasite. I mean, it's borrowing the coats of other viruses in order to get itself around, which is a very clever strategy. I mean, that's something evolved to do that.
Well, there are other viruses that do that.
Oh, yeah.
So the adeno-associated viruses, if I'm not mistaken, can use either adenoviruses or herpes viruses to prevent helper.
But in that case, it was the helper is a polymerase, not a glycoprotein.
Right. So they're probably, and of course, we don't know these actually infect people yet, right?
Right.
This is an experimental construct.
Yeah. This is all experimental, and the big question is this happening out in nature?
Yeah. And the first place to look would be in Hep C patients.
Mm-hmm.
I think so, yeah.
Mm-hmm. Gosh, I wonder if anyone... I mean, I'm just stunned that no one did this experiment. It's so simple, right? And secondly, why hasn't anyone looked in Hep C patients? But there are plenty of samples that are available, so that can be done quickly. It's probably already done. It's probably submitted.
Yeah.
Right.
We're going to be doing it in 2 weeks.
Stay tuned.
So what they say here is very interesting in the discussion. HDV can persist in the liver in the absence of HBV for at least 6 weeks.
Wow.
In the mouse.
In people.
In people.
In people. That's really interesting. So the RNA is just there replicating in cells, right?
Right.
It's not making particles. So it can last at least 6 weeks. So if that person gets infected with another virus in the meantime, it could potentially escape. Now, it'd have to be a liver tropic virus. So Hep C is a great candidate. And of course, other viruses can infect the liver as well. But not West Nile, I believe, right, Dickson?
I don't think so. No, it's mostly a central nervous system thing.
Well, it also infects,
mosquitoes,
immune cells, lymphoids, like dendritic cells, and
then it infects amphibia. It's one of the most promiscuous infectious agents known to humans.
And once the hepatitis D gets out of the liver, then then it could catch up with any of these other viruses that are in other tissues, right?
Presumably. Mm-hmm. So what happens? Don't liver cells die periodically and release their contents into the circulation, right?
Sure they do.
So it could happen elsewhere, not just in the liver, maybe. Yeah?
It's possible that the Kupffer cells play a role in preventing them from getting out, though.
So if the RNA is taken up by some immune cell, which is then infected, could be interesting. So lots of people need to now look in people for hepatitis delta, independent of Hep B.
Yeah, they might want to look in dengue patients too.
Yep.
I was double checking to see whether dengue replicates in the liver because I couldn't remember, and I'm finding evidence that it does.
Right.
Really?
In hepatocytes or just some immune cells flowing through?
This says hepatocytes. It's a paper from Ava Harris.
Be darned.
Interesting. Very interesting. So let's everyone as this arc will continue.
Yes.
For a
and
while.
by the way, all the papers that we have discussed on the show today are open access.
Open access.
Yep.
What do you think of this one, Kiki? This you don't have to say anything because I didn't teach this to you, so it's okay.
I'm just wondering if Hep D has a tropism on its own, or if it actually is just.
On its own, it's just an RNA. That's the thing. It depends on what capsid it's encapsulated in. And historically, we've thought it's only Hep B, so it would be liver tropic. But now it could be anything, maybe.
So is it just it's cooperative with Hep B?
Oh, well, that's a good question. So the general thought is that it makes disease of Hep B worse.
But what about these like VSV that I talked about?
It doesn't interfere. It doesn't make it better. It doesn't help. So it doesn't.
So it's neutral.
It seems to be neutral. Yeah.
To follow up on what Alan was saying before, Vincent always says that he never met a virus that he didn't like.
It's true. Of course, I haven't gotten really sick yet. Who knows? But I would understand.
I don't think I've met a pneumovirus.
Well, window and influenza too, but that's another story, isn't it?
I think this is just fat. Both of these topics are just so cool.
Yeah, they're great.
This delta is, and now, okay, that's I thought about what I wanted to say. In snakes and birds, makes sense now. Must be some other virus
With some other virus.
that is complementing. So not too long ago, Delta viruses were found in snakes and birds. No Hep B. So what's going on? Here it shows it could be some other virus.
Well, West Nile virus does infect both snakes and birds, by the way.
Okay.
There you go.
So West Nile's a candidate.
Sure.
A good candidate. I mean, yeah, I would say so.
So some birds it makes very sick, right?
The corvids, the crows and the blue jays.
Can you make your crow sound? And
it's a murder.
In snakes, what does West Nile do to snakes, Dickson?
I have no idea. It kills the corvids, but it doesn't kill a lot of other birds. So I think that the snakes are probably not deeply affected because their body temperature fluctuates so much with the environment that the virus probably doesn't achieve very high levels. But in birds, their body temperature is higher than ours, so I think the replication rate is higher. That's why they're more susceptible to dying, I think. And in horses, I must say that horses are the most susceptible mammal that we know of for lethality effects of West Nile. Mm-hmm. So it's a big problem out in the West.
So I was very happy to see in this paper they called it hepatitis delta virus, not satellite. It used to be called satellite, but that's been now considered derogatory to viruses because it's a virus.
Do horses have their own hepatitis virus?
Yeah, they do.
That's what I thought. Okay, so this could very well prove to be very serious. Horse racing in the world is very
lucrative.
Sure.
Dollar intensive, shall we say.
Well, that's why we have a horse West Nile vaccine.
Yeah, exactly.
It's
also one world hypothesis sort of stuff. It's cheaper.
Very good. Yeah. Where did you learn that?
I don't know. Right. Some class I took once.
Very good. One world. There you go. Very exciting. Okay, there you go. Hepatitis delta, polymerases that don't need primers. What is this world coming to?
Yeah, really.
Or where has it been?
Or more likely, where is it going? Let's read some email here. I have an email from Stefan, whose subtitle was one of 5 North American postdocs. Dear TWiX Cooperative, thank you all for your continued commitment with the public outreach of science education. All the TWiX podcasts, including Immune, serve as an exceptionally valuable resource. Each has its own character, but what I especially enjoy is the overlap of topics across the different fields of virology, microbiology, parasitology, evolution, and immunology. The incredible stories of co-adaptation are certainly my favorite topics. The deeper we look, the more we see, much like astronomers peering into the deepest recesses of the universe.
Right. That's a great analogy. That's a great analogy.
It was fabulous to hear Kara on last week's TWiV talking about the great work being done by her lab. I was a guest on TWiV 194 back in 2012 at the Madison ASV with 4 other postdocs, Kara being one of the others, along with Matt Doherty, John David DeJong, and Helen Lazier. Yeah. It is unfathomable that 7 years have passed since we all got to speak with the virology podcast guru Vincent. It was a wonderful experience. Kara was correct. I'm still at Stanford in Professor Ann Arvin's lab, where I'm now a senior scientist. I've been having way too much fun with my research, investigating the molecular mechanisms of varicella zoster cell-cell fusion and its relationship to pathogenesis. My position gives me opportunities to work with postdocs and train them in some nuanced techniques to dissect the molecular mechanism behind cell-cell fusion caused by VSV. I also have wonderful opportunities to pursue my own ideas and learn new techniques. In recent years, I've turned to structural biology approaches using cryo-EM. I'm in the final throes of wrapping up my first structural biology manuscript that uses cryo-EM to investigate how a human antibody neutralizes VZV. It would be wonderful for the 5 postdocs from North America to reconvene with Vincent in the not-too-distant future. Sadly, I'll not be able to attend the ASV's excellent conference at the University of Minnesota this year, as it coincides with the International Herpes Virus Workshop in Knoxville, Tennessee. It's a shame that the 2 meetings overlap this year, but you can't go to all the conferences all the time. Please keep up the outstanding work that all of you do to keep the podcast going. I'm a big fan of the longer podcasts, but I fully understand that time is a precious resource for you all. However, I, for one, would never complain that a podcast is too long. I'm writing to you from a rather overcast La Honda, California, where it's currently 15 degrees Celsius. Being British, I can't help but talk about the weather. It's a great way. It's a great way to start any podcast. Don't listen to the naysayers. Is that true? Do the Brits like talk about the weather?
They do, of course they do.
Best small talk they have.
It's the best small talk. Oh, God.
And they've got weather like you wouldn't believe right now, by the way. They're getting rained on like crazy over there. It's just amazing.
You mean it's gone beyond the drizzle?
Lots of flooding. Yeah, way beyond a drizzle.
Wow.
The best quote I like about British weather was from a series a long time ago, Alistair Cook's America.
Oh, yeah.
Where he talked about a headline in a British paper about the weather that said, 70 again today and no relief in sight.
Oh, my gosh.
Yep.
Brianne, can you take the next one, please?
Of course. Adrian writes, Dear TWiV team, my Facebook is full of warnings about reduced measles immunity in adults who were vaccinated in the 1960s based on stories like these. And she provides a couple of links. A family member is asking me, is there evidence that immunized 50-year-olds are getting the measles or have low seropositivity? I have access to journal articles and have found very little on measured seronegativity or infections in this age group. The one article I can find shows most measles cases in adults are likely in unvaccinated individuals. Is there a journal article that provides experimental evidence that middle-aged people are at greater risk? I appreciate the help.
So I looked up this, and there are a couple of studies. But remember, we only started vaccinating in 1963, so we don't have really old people.
Right.
Just, what is it? So 63 would be a 56-year-old, right?
Mm-hmm.
And so these papers that I found, they show there is some waning of immunity after vaccination. Of course, after real infection, it's lifelong. But after the vaccine, which is an infectious vaccine, it does wane. Now, the one article she provided, the Times article, is interesting because it shows in the beginning there were a couple of different kinds of vaccines used, and only the one we use today was settled on later, and it seems to give the most durable immunity. And initially they were using inactivated vaccines, for example. So I think, if I got those first batches of vaccine in the 60s, I would consider getting immunized. There's zero downside, and with the measles outbreaks currently, you don't want to risk it, right?
Yeah.
So that's what I found.
I've got a regular checkup coming up soon, and one of the things I was going to ask my doctor about is, should I get a booster measles shot?
That'd be interesting.
I'm just going to stay out of Brooklyn.
Well, you got to stay out of Rockland County, too.
That's true. That is true.
But anyway, these papers do, and I'll put the links in the show notes, they do show it wanes. The immunity wanes like about 3% a year. So you can imagine as you get further and further out, you're going to have some. And what's interesting is that the mumps component of the MMR vaccine, that wanes, and there have been some mumps cases, especially on college campuses. There are often mumps outbreaks, and they're due to waning mumps immunity. Mm-hmm. So
yeah, there's a lot of good data on that.
Yeah. There's one, where did someone tell me? There's just one. I can't remember. So there's always one somewhere. Okay. Rich, can you take the next one?
Sure. Fisto writes, G'day, Team TWiV. TWiV 539 multipartite plant viruses on why 8 separately packaged viruses would need to complete infection. I have an ill-informed hypothesis, he says. I think the key to why the group approach may be selected for is in the delivery method. The aphid vector will always deliver a cocktail of whatever virions are in the xylem, so multiple variants of a given virus will reliably be delivered all together. Then let natural selection have its way over time. The same 8 variants of the virus repeatedly meet in a new host, each with an individual trick up its sleeve, but none with a dominance that could exclude the other 7, given the selective pressure towards small particle or genome size. Duplication of function would also be discouraged, leading to a selective loss of genes from as many as 7 of the 8. It would make sense to specialize each of the 8 variants performing the trick it does best, knowing that the other 7 will perform the other tasks needed for eventual replication and transmission of all 8. It's a kind of viral symbiosis. I like that. Where the colony performs better than the individual. Each of the 8 sees the other 7 and their products as just a given property of the host environment. The exclusion from any given infected cell of further infection by one of the 7 other virus members is to be expected, so as to ensure the ensemble functions. Each must claim some turf in the host for the system to work as a whole. Feel free to shoot my hasty hypothesis down in flames. I have a day job as an IT consultant, so it's okay.
Allow me to interject one small quibble, and that is the word symbiosis probably should be a synergy. It would be a better word,
Okay.
I think.
All right.
So symbiosis is the relationship between 2 dissimilar organisms,
Right.
whereas these are all viruses of the same
Right.
kind. So that
There's a wonderful postscript, too.
P.S. In Adelaide, Australia, excuse me?
no, go for it.
In Adelaide, Australia, it is now a chilly 13 degrees Celsius, or that is, I think, 56 foot gallons in USA.
Yeah, that's about a right conversion.
I think that's great.
Maybe a 52
Foot gallon.
I think his hypothesis is fine.
Yeah.
Makes perfect
Yeah.
sense because plants are weird that the cells all share everything, right?
Yep.
Right.
So you could imagine viruses evolving where everything is packaged separately and it works. Yeah.
Yeah.
You're doing a good job there.
Yeah.
Visto.
Foot gallons.
Foot gallons.
Love it.
Once foot gallons.
I like that.
It's a new measure of.
Yes.
You know, Dickson, over on TWiV, we have some listeners in Adelaide.
We do.
It's interesting. Alan, can you take the next one?
Sure. Cal writes, hello, writing from Lothian, Maryland, outside Washington, D.C., where it is still warm, 79, 26C. It's at 9 p.m. Summer has arrived. I'm a toxicologist and risk assessor at a federal agency in D.C., not a microbiologist, a couple of immunology courses while in grad school, but I volunteer at the Smithsonian National Museum of Natural History, and I had the privilege of being trained on Outbreak. I got turned on to the TWiV group of podcasts by a neighbor who somehow knows Alan Dove. Cal, let me know who that is. I didn't know any of my friends from Maryland actually listen to the podcast.
You have many admirers, Alan. Many.
I guess. I only understand maybe 25% on a good day, but I can generally follow along and always learn something new. Listening to back issues, I heard Stephanie talk about The Pregnant Scholar. I wanted to alert you to the book Motherhood, the Elephant in the Laboratory. Full disclosure, I'm a contributor, but I made and make no money from this project. The book was conceived, good choice of terms, and edited by Emily Monoson, and it contains collections of women's stories organized by decade so you can see how things have and have not changed. Worth a look. Also, for the TWiV folks, you might find Emily's most recent book, Natural Defense, Enlisting Bugs and Germs to Protect Our Food and Medicine, Island Press 2017, to be of interest. Thanks for the hours of entertainment and education.
Very nice.
You're quite welcome, Cal.
Nice. Well, it's kind of near where you grew up, right? Maryland's near Delaware.
Yeah, that's at DC, Annapolis, Baltimore area. I grew up right outside Annapolis, and so it's probably an old friend from high school or something who lives in that area. Could even be a relative, but
Could be.
yeah, Cal, let me know.
That's pretty funny.
Yeah.
I didn't even realize you were in that email. It just came up. Your turn.
There I am. Yes.
And let's do one more. Dixon, can you take?
I'll try.
It's a little complex. Okay.
Well, it's Anthony writing to Martin, so we've got to. We're publishing their.
So let me just remind everyone that Martin Beer was on TWiV 548 in Rotterdam. Okay.
Right.
Okay.
So it's entitled Anthony wrote to Martin Beer about TWiV 548. In TWiV 548, there's a discussion of bornaviruses and transplants, and he gives a reference for that. In the podcast, there's mention of shrews, squirrels, and rodents. Perhaps it's my personal confusion, but it seems to me that it's being said that both shrews and squirrels are rodents. Though at a glance, they might look like
that would be wrong.
Shrews are insectivores. And he's correct in that, of course, and he gives a reference for that one. And have no close relation to squirrels, mice, or other rodents. If my reading is correctly, if I'm reading it correctly, the paper includes horses with shrews as possible sources of the virus. There's no chance that the transplant donor was infected by a horse? That's an interesting question. I found interesting your mention of the pet cat. I wondered if cats allowed to roam serve as the last mile in the transmission of some emerging diseases, with the exception of young boys, speaking as one who many years ago was once bitten by a bat infected with rabies. Few people are motivated to interact with bats, shrews, moles too, or rodents, particularly animals behaving erratically. The same is not true of cats, who after their diversion will return home and share their owner's bed. By the way, I share a similar situation. I was bitten by a rat, a wharf rat, and I'll never forget the experience. Although it didn't have rabies, it was certainly behaving abnormally. Thank you, Anthony Ozlowski. Martin's response. Dear Anthony, many thanks for your email. I will try to clarify the different points. Mammalian bornaviruses are only transmitted by their reservoir hosts, squirrels for VSBV1, shrews for BODV1. The transplant transmission seems to be a very unique exception. Shrews are insectivores, and no rodents' accidental hosts like horses, sheep, or humans do not further transmit bornaviruses. We call them, therefore, accidental or dead-end hosts. Infection from a horse is there, to our knowledge, not possible. The role of cats is also for the transmission of bornaviruses still a hypothetical scenario. Further studies are therefore necessary. Please do not hesitate to contact me again if there are further questions. Best wishes, Martin.
It's a very interesting story. We have to listen to TWiV 48, but they we talked about variegated squirrels, which are apparently bred in parts of Germany. They're exotic pets, right? And they're breeders. And we talked about a paper where a couple of breeders got actually bornavirus infection from them. So they're accidental hosts. But the variegated squirrels are okay. They have lots of virus and they shed it and they're fine. They have lovely stripes on their backs, by the way. That's why they're prized.
Or they're variegated.
And that too. That's
Indeed.
And the other story of Martin's was they had a organ donor give organs to 3 different people. I think he had died in a car crash or something. And all 3 people got a bornavirus infection. 2 of them died of encephalitis.
Oh, my.
And so somehow the donor had gotten infected, and they don't know how. But these viruses are resident in shrews and squirrels throughout a certain part of Germany. And that's why Anthony says, could it come from a horse? Not likely, because a horse is a dead-end infection, just like humans are. So it's a really interesting story. He talked about that on TWiV 548. Okay, let's end up with some emails. Not emails. Let's end up with some picks of the week. And you should think of a pick, Kiki. You know what a pick of the week is?
Favorite
something
article?
Cool that you want to. It could be anything. It doesn't even have to be science related, but it would be nice. But we'll give you some time to look stuff up on that, computer you have there. Brianne, what do you have?
There is a virologist named Ephra who I follow on Twitter. He does a lot of really great microscopy. And recently someone told him that he shouldn't try to be unique as a scientist, that made him useless. And as a result, he actually started a website called Unique Scientists, where people are able to put up their profiles and show the diversity of people who are in science and all the people who are represented as scientists.
How nice.
Very cool.
So it's a really nice way to sort of show what scientists really look like and who scientists really are. And he's trying to have a lot of people submit profiles and show who we are. So I think that it's a fantastic response and a great initiative altogether. I think people should take a look at it.
I think that's great.
It's cool. I like Ephraim's tattoo. He's got mitosis on his arm.
Yes, he does.
That's great. So there's another one I saw recently. You must know where this is, Brianne. Someone said, put up your stories of feeling like an imposter, a science imposter. Did you see that?
I have seen them. Yes, I have.
This week I felt like writing one. Especially after our faculty meeting here yesterday, I felt like an imposter.
No, that was me last week.
Sometimes I just don't feel I'm in line with the rest of the faculty here anymore. You know what I mean?
Too much ahead of them?
No, I'm not ahead of anybody. I'm just doing different things. And more particularly, they don't get what I do at all, right? So
You got to listen to the podcast.
Never.
Well, Ephra has some T-shirts as well. One of them has a B on it, and then it says unique, so it's supposed to be be unique. But
cool.
I'm looking forward to receiving in the mail. I ordered it.
Cool. Alan, what do you have?
I have a cool little app that I downloaded a little while ago. It's called Seek by an organization called iNaturalist. And iNaturalist is it's a place you can sign in and post observations from nature. But this app, you put it on your phone and then you walk around and it's Pokemon Go for real life. So you point it at some species and it identifies it.
Oh,
what?
I'm so ready for this.
That's amazing.
Including plants, right?
Long,
wow.
Including plants. So, I was like, oh, okay, well, maybe it works, maybe it doesn't, but I'll give it a try. And I downloaded it, and of course, I'm sitting at home, so I turned it and pointed it to the cat. And one of the cool things about it is you can see it thinking. So it immediately said mammal.
Ooh.
And then the cat kind of turned her head a little bit, said carnivora, and then cat.
Wow.
Domestic cat.
So it's taking pictures, right?
And then
Does it take it down to
pictures.
species, Alan?
You can log that, and it gives you badges for logging a certain number of species in different kingdoms and phyla and all that. And I went outside. I did a couple of plants. Plants, it's interesting. The more extreme they look, the easier it is for it, obviously, to identify. So you point it to a random shrub, and it says dicot.
Right.
That's about as far as it gets. Because of course with plants you have to have the flower structure or all that. But Japanese maple it got immediately. Things that are a little more extreme, peonies, it figured that out because they happen to be in bloom now.
How about insects? Did you try any?
I have not yet gotten it to because the insects move away, and same with birds.
Well, there's a solution to that.
Yeah, exactly.
There is. Audubon
involves a
found
pin.
that solution.
So I think
Alan, this has huge application to trout fishing.
Yeah,
sure.
Well, you want to identify a trout? You know what it looks like.
No, to identify the insects that are hatching.
I see.
Yeah.
This would be fabulous.
It's very cool.
I just downloaded it.
Yeah, I'm downloading it right now. Alan, did you point it at yourself?
Yeah, right.
Right after
Primate.
that. Yeah.
What if
It says unique scientist.
No, it comes.
I can see that you were walking around outside, Alan, using this a lot now, right?
Wow.
When I remember, I'll whip it out. Oh yeah, what is that? And now
So not too long ago, you did a similar app for stars and planets, right?
yes.
So last week I was at Princeton working on the textbook, and one night we were all sitting outside and talking, and I look up at the lower part of the horizon and at the low sky, and there's a really bright something there, and nothing else is out. I said, that must be a planet. So Glenn Rawle whipped out his app.
Jupiter.
And he said, it's Jupiter.
That's right.
Yep.
It was Jupiter. How cool. So someone said, is there one for plants? And I said, well, I don't know. So here, I'm going to send them this one.
No, but there's one for planets. So Jupiter was so close the other night that they claim with a pair of good binoculars you could see 4 of its moons.
Yeah.
Yeah, they're very bright. Yeah.
In addition to the disk of Jupiter and maybe a band of cloud.
Rich Condit, what do you have?
I have 2. I'm going to claim 2 here. One is a pick and the other is a news flash. The pick is a New York Times opinion piece called "Want to See My Genes? Get a Warrant." And it's basically an op-ed about the ethical issues involved with police using DNA to solve crimes. All right. What I really liked about it was one of the things I liked about it was a particular case that it focuses on where there was an assault in a church. And part of the evidence they collected was some blood that pretty obviously, given the circumstances, came from the assailant. And They got DNA sequences on the blood and sent it to a publicly accessible database called GED Match. And they didn't get a match on it, but they got a fairly close match that they figured must be a relative who lived several states away, but then did some investigative work with that person and found that there was a cousin, a nephew with a child that lived in the area, and were able to track it down to a 17-year-old high school student, got a DNA sample from that student, and it matched the original blood. Okay.
The creepy part is how they got that DNA.
Yeah, right. A school resource officer watched the student during lunch at the school cafeteria and collected the milk carton and juice box that he'd thrown in the garbage.
Wow.
This is
So
not
creepy.
just sequencing yourself. I know that people get caught a lot nowadays with relatives who sequence their DNA for like any of the Ancestry.com sort of things.
That's so creepy. This guy's going to school and
Yeah, that's a little odd.
So this apparently a similar technique was used to solve a crime with the Golden Gate Killer, the serial killer in California some years ago. But at any rate, this article, raises ethical issues, and the title sort of says it all. You want to see my genes? Get a warrant.
yeah, it's really good. It really brings up a lot of stuff because apparently they're now doing it for all kinds of incidents, not even crimes. They're just,
right. So what are the
It's amazing how much they can find. My mom is actually adopted and used similar databases to find her birth family. And it's sort of amazing how all of that can be done now.
Yeah, actually, I heard a story recently of somebody who, submitted their DNA, and basically it came back that it identified a father that was not her family father. Okay. And it was like, this person was like 53 years old, and she was able to contact her bio father, who she didn't know existed. Okay. And the response from this guy was, I've been waiting 53 years for this. Okay. I really wanted to know my daughter, but I didn't want to get in the way of her life. It was, and it was a great story. Great story. At any rate,
that wouldn't necessarily be the ending of that story.
No,
right.
No, it wouldn't necessarily
Your mileage
work.
may vary.
It doesn't have. That's right.
It's the
beginning. Have a happy ending. At any rate, while we have been podcasting, I got a text from my daughter pointing out to me a Washington Post article from a few days ago titled, NIH Director Will No Longer Speak on All Male Science Panels. Okay, so this is Francis Collins announcing that he will no longer participate in any panel that is all male, which I thought
he is no longer doing manels.
Panels,
as they're called. And I saw this on Twitter. It was like, okay, yeah, that's good.
Yeah, but it's got to go beyond him.
Right, but it's a very strong statement from somebody who gets invited to a whole lot of panels and who sets the example for others in similarly situated to say, look, this is not okay for conference organizers to put together an all-male panel on really any subject and think that that's just fine in 2019.
Yep, totally. It's good. It was also in the Times.
Yeah.
Dickson, what do you have?
Well, I watched a show last night that I'm sure you all were aware of, but maybe didn't say anything about. It was Nova, and the title of it was Vaccines Calling the Shots. And it was a summary of the current status of the situation that we now face, and interviewed lots of distraught parents, distraught physicians, pediatricians, people with children. But one was very, very touching story of a little kid who had whooping cough, another mother whose child died from a disease that could have been prevented with a vaccine. I'd highly recommend watching this. It's written, it's performed at a level that everyone can understand. There's no doubt about what's going on. There's no misunderstanding at all as to what vaccines can do and what they can't do. They brought up the fact that, 1 in a million, there'll be an adverse reaction to the vaccine. But is that any better than 1 in 1,000? Goddamn right it is. And we keep saying that, but this show is watched by many, many, many people. Yeah,
more than TWiV, sadly.
Yeah,
I'm sorry to say that, but it's true.
Yeah,
but they're doing good stuff.
It's good. In fact, if you go to the page at the bottom, it says "Explore More," and they have another 10 videos about vaccines.
Exactly.
Which is like Immunity and Vaccines Explained: A History of Vaccination. So it's good. I think it's great. But unfortunately, there are still wackaloons out there.
You know what? It doesn't matter how logical and how correct you are, people will still disagree with it.
Of course.
Yeah, I mean, there was a article yesterday that came out with a couple celebrities and big names, and I think Kennedy or something
So that's
in California. California saying that vaccines are based off that phallacytical 1990s paper that they're bad for you and cause all these diseases still.
So my 2 picks are all about that. So they're both New York Times articles. One is New York State is about to eliminate religious exemptions for vaccinations.
Good.
Woo.
Which I think is great. It follows a few other states, California, Arizona, West Virginia, Mississippi, and Maine. I think
Mississippi?
Massachusetts doesn't allow religious exemptions, right, Ellen?
I didn't think they did.
Yeah,
when I saw that list, I was very surprised to see Mississippi on it. But I mean, it's possible for something progressive to happen anywhere, but that was not the place I expected.
Yeah,
I'll bet you there's a different reason for it too.
Yeah, probably yes.
And so Cuomo signed it. Yeah, it says it joins these other states. And Assemblyman Michael Montesano, a Long Island Republican, framed the bill as an attack on people's First Amendment rights. It's up to the parent to decide what happens to their child.
And who they can infect.
Right.
It is not. It is not your right to infect other people.
Correct.
Which is a lecture quote from our semester this past year.
Really?
Yeah, 3 times.
No.
It's an important point. It's an important point.
Listen closely.
I tend
Chief
to
medical
say
officer for New York, Howard Zucker, is on television now.
Yeah.
Actually standing in front of you, looking you right in the face.
Why are you watching TV during TWiV, Dickson? I'm just kidding. It's okay.
I have 2 eyes.
You can do whatever you want. You're absolutely right. So I think you have your right to your religion and your God. That's absolutely fine. But you do not have the right to infect other people.
That's correct. Or endanger the lives of your own children.
And so my other pick is also a Times article, which is the story that Kiki mentioned. Jessica Biel weighs in on vaccine fight, drawing fierce pushback. And so she, with Robert Kennedy, famous anti-vaxxer, which they say has spread misinformation about vaccines. It's absolutely true. They all spread misinformation. But she said, what did she say? She likes vaccines.
She showed up at an event. They're lobbying against a more stringent measure in California because California passed a rule that they would getting rid of religious exemptions, and so now the only way to get out of your childhood vaccines is a medical exemption. But people have subsequently gone and found doctors. There are a handful of doctors who are of this same counter-rational belief system who will write these exemptions. And so now California wants to make that more strict, that the exemptions would have to be reviewed and approved at the state level.
Great. It's great.
And Jessica Biel showed up at this event opposing this measure, saying she's not against vaccines, just that it's about parental rights.
Yes.
And no, it's not. Yeah. No, it's not.
No. You don't have the right to do this.
It's not. And these are constitutional
You do not have the right
rules.
to abuse your child. That's a matter of settled law.
That's correct.
Yeah.
I mean, it's incredible because there's a conflict here between the people that are against abortion because they think that the fetus has a right to life. And then they don't think that they have a right to the vaccination. That's crazy thinking. That's just absolute bonkers.
Someone wrote to her on Facebook, go to med school, do some research, and then maybe you can spew your harmful, baseless opinions. Meantime, keep your kids away from mine. Well, at least that's a pro-vaccine. So there's a good quote here from a physician that I wanted to read. Let's see. So Tara Smith is quoted. She's a microbiologist at Kent State who I know. I think when Jenny McCarthy was the main face of the anti-vax movement, medical professionals were a little dismissive. Then she built this empire around mommy warriors, became the face of women who felt they were empowered by their personal experience with their children and what they had seen of vaccination.
What had they seen of vaccination?
Just their own experiences, N equals 1. That's the problem. And there was some other physician here who said it's a lot of malarkey, these anti-vaxxers, which I always like seeing that. Anyway, I just don't understand. There's another article in the Times this week as well. I think my other pick also explored this, why people feel this way. People say, well, the science is all behind the times. We've done our research. We know that vaccines are problematic. And I'm just saying, folks, you're not reading the literature. You're reading crap online, and that's not what you have to do. And I understand the literature is hard, but the other stuff isn't right. Science is not behind it. And
what you read online, especially if you're using Google, is driven further and further in whatever ideological direction you're reading because of Google's algorithms.
Yeah, it's an echo chamber.
Exactly right. Just try ordering something on Amazon, and the next 15 months you get pop-ups on all those items that you didn't buy.
Yep.
It's just, weird. It's weird out there.
I know. This is extremely discouraging that there's so much anti-vax sentiment. And I think in one of these articles, they're saying it's becoming a entrenched sentiment of the left, right? Yeah. Which is really troubling.
Just sad.
But I'm glad some states are doing things. It's really good. Yeah. So we'll see where this ends up. Okay. Kiki, you have anything for us?
I was sent something by a friend, two friends, and then by James Hetherington as well. And one of the ones I just got sent, which I like his, is that they have found a way to convert type A blood to type O blood.
Oh, yeah.
Did you just crowdsource the pick of the week? You just ask your friends for a pick?
Yes, I did.
Wow, that's...
I see it as an evolved way to do it.
Oh, that's great. That's a good idea.
Excellent.
Where was this?
So this was referenced on Popular Science, and then Popular Science has a Nature Microbiology paper.
So type A to O, is that the conversion?
Yeah. Enzymatic pathway in the human gut microbiome that converts A to universal O type blood.
How cool.
Yeah. Which will help a lot for all types of blood donation.
Yeah, that's cool. I thought you were going to pick the article where they made cannabis and yeast. Did you know about that?
No. What is that one?
They put the entire metabolic pathway into yeast to produce, and not the active ingredient, right?
THC.
Yeah. I'm saving that paper for TWiV sometime.
I've heard that referred to. I've heard that referenced as baking bad.
Yes.
Woo!
That's great.
Not mine. I'm sad to say I did not come up with that.
Baking bad. You can bake it into bread, right? I'm looking
beer out of it.
Beer?
Beer.
Beer is fine as it is.
Yeah.
Right?
It's even finer.
Scientists brew cannabis using hacked beer yeast. I like that very much.
There you go.
We have a listener pick from Justin. He sends us an XKCD comic, and he writes, "Don't forget the mouse over text," and Alan Dove, I'm sure, can explain. So, Alan Dove, what?
Mouse over text is pretty easy to explain. You hold your mouse over the cartoon, and some text will come up.
No, it's not doing anything.
Oh, okay. So the strip is about an apple a day, and it's a press conference where a scientist is saying an apple a day keeps the doctor away, or at least it used to, and then talks about how doctors are becoming highly resistant to apples, and
I see.
Still work on most of them, but we don't know for how long. And then the mouse over text is even the powerful tart Granny Smith cultivar is proving ineffective against new gram-negative doctors.
Oh,
that's good. Yes. I was mousing over the map at the top, which has nothing, which is different. Now I got it. I don't know why we needed you to explain that, right?
I don't know.
Thank you, Justin. All right, that is TWiV 552. So our snippet was longer than the paper, right?
Yes.
Yep.
It's because there were two snippets.
Both really cool.
It doesn't matter, really.
Nah.
It doesn't really matter.
Just
all
words.
just interesting stuff.
We can just do what we want.
In the end, it doesn't matter. Yeah.
You can find all the show notes and links at microbe.tv/TWiV. And you're probably listening on a phone of some kind. Do you listen to TWiV?
Yeah.
On a phone?
Yep. Podcast app.
Apple Podcasts, right? So you've subscribed to TWiV, right? So you should emulate Kiki here, who has subscribed.
Highly recommend it.
And just subscribe so we know how many people are listening. And you'll like it, really. It's good. It's good stuff. Even if it's complicated, don't worry about it. I listen to things I don't understand all the time. I listen to this this podcast about computer security. I don't know half of the things they're saying, but I listen because I figure I'll figure it out at some point, right?
And you'll sound cool at dinner conversations.
Yeah.
Well, I think you're cooler with viruses, frankly.
Oh, that's what I'm talking about.
Oh, okay.
TWiV.
That's good. That's right. And if you really like what we do, consider supporting us. You can go to microbe.tv/contribute. And there are a number of ways you could help us. The simplest is Patreon or PayPal. You can give $1 a day. No, $1 a month. Sorry.
$1. Hey,
You could also do $1 a day. Yeah.
Don't say no to that.
Actually, some of you actually do give $1 a day.
I hear $1 a day keeps the doctor away.
And we're very
Yes.
grateful
There
for
you
that.
go. There
But
you go.
any amount you can give $1 a month is, I think, the minimum, which when you think about it, you can't do very much for $1, right? You can't get a cup of coffee. Kiki, can you do anything for $1 in New York City?
I think dollar stores are even more than $1 nowadays. So,
Oh, the dollar store. I
true.
forgot
Yeah.
about
It's
that.
true.
but I think with tax, it's like $2.
Okay. So microbe.tv/contribute. And of course, questions and comments. We love them. We love your questions and comments. Even if you just say, hi, I'm so-and-so in the weather here and I like listening. That's fine. TWiV at microbe.tv. Dickson Despommier is he's an emeritus professor here at Columbia University. He's at home today.
Mhm.
And but you can find his work at DespommierPhotoArt.com, which is a cool site, and ParasitesWithoutBorders.com. Thanks, Dickson.
Pleasure. A pleasure.
Brianne Barker is over at Drew University in New Jersey, and she's on Twitter as BioProfBarker. Thank you, Brianne.
Thanks. It was great to be here.
I will see you in vivo next weekend.
Yes, I'm looking forward to it.
Don't forget,
I won't forget. Don't worry.
we have a TWiV. It should be fun. At ASM Microbe, San Francisco. When are you going out? Thursday?
I'm going out Thursday.
Okay, see you there.
I will see you.
Rich Condit is an emeritus professor, University of Florida, Gainesville, currently residing in Austin, Texas. Thank you, Rich.
Sure enough, always a good time.
Had a visitor today. So Dennis Benty is a virologist in Galveston. He runs the BSL-4. He's a virologist. And he came with a student and a postdoc. And the student knew you. She had grown up around Gainesville. She knew you and McFadden.
Oh,
How about
You got
that?
a name?
Small world.
you got a name?
No, I forgot already.
Okay.
Sorry. I have a picture, though. They wanted to come here and see the Wall of Polio. They're actually visiting someone else.
Wall of Pope.
And my colleague, Ann Moscona, she came down. She says, you have some fans here. They want to come see you. Is that okay? I said,
Send me the picture.
they wanted to have a picture in front of the wall. Yes.
Post it on Slack.
Yeah, that's a good idea. We started a Slack channel.
Yeah.
You know what Slack is, Kiki?
Managerial app.
What's that?
Managerial app. That's how people get work
managerial
done.
app. Do you use
That's how my startup brothers do stuff.
Oh, yeah?
Yeah, both my brothers are tech startup people, and they Slack
Really?
it all day.
Where do they live?
One lives in San Francisco. His name's Thompson, and he does a educational tech startup. And the other one's Paul, and he lives between New York, San Francisco, Singapore, London, and Paris, depending on the day of the week.
Pretty cool. Wow. I like tech startups.
Yeah, he does blockchain.
Now, what is blockchain?
That's too long a question. Come on.
Yeah,
It's too long.
Yeah,
Is it a way of making
first.
financial transactions secure?
It's a way.
not
of
just financial
secure-
transactions.
It's a way of securing anything. It's a way of securing data.
on the
that
on the
is-
internet, right?
Yeah, digital data, and it's very difficult to hack because the security is distributed rather than using a single trusted source.
Okay. But it takes a
So-
long time to
Yeah,
use it 'cause-
but it can be useful for medicine. So they're looking at maybe putting the electronic medical records into a blockchain type thing. There's a couple projects in that.
It can be used to secure any kind of data.
Yeah, I hear that over and over on my security podcast. I never know what it meant because I'm usually driving and I can't look it up. Right? Alan Dove is at turbidplaque.com. He's on Twitter as Alan Dove. Thank you, Alan.
Thank you. It's always a pleasure.
And our guest in studio today has been Kiki Warren. Did you enjoy?
Oh, this was so much fun.
Like, where are you from? What should I say? Say everybody else. Are you on Twitter?
I'm not on Twitter. I'm on Instagram.
What is your name on Instagram?
Just Kiki Warren. If you guys want to follow my exciting stories with my dog.
Kiki used to bring her dog to lecture.
Yeah, Bernedoodle named Knox. She is very fluffy. Name. Yeah, it means night.
Well, thanks for coming.
Yeah, thank you guys for having me. This was a very enjoyable Friday.
Good luck with your medical school thingy.
Yeah, we'd love to follow you through your medical career.
Yeah.
Hopefully, I can come back sometimes. I'm 2 weeks out from the MCAT, so.
No, you're going to study.
Oh, this was studying. This is fabulous.
Well, good luck with that. You got any summer plans? Are you working?
Possibly at the lab nearby here.
Oh, come by. You can come again.
I would be honored.
Yeah, sure. Anytime. I thought you were going to talk more. I'm glad you didn't. No, just kidding. Just kidding. Just kidding. He used to talk a lot on our walks to the subway.
I have so many questions, but it would fill the entire space of this entire podcast.
You ask a lot of good questions. It's good. I mean, after class, you would come down to the lecture, and someone else would ask a question, and you would just jump in and start talking, right?
I would answer their questions, though, a lot of the time. So.
I'm Vincent Racaniello. You can find me at virology.ws. I want to thank ASV and ASM for their support, and Ronald Jenkins for the music. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.